Here's a truth that might surprise many. Impaired energy production in type 2 diabetes could stem from a reduced ability of mitochondria to effectively process ketone bodies. But here's where it gets controversial. Often, when people think about boosting ketone levels, they assume the body's cells will automatically make better use of them. However, recent research suggests that merely increasing ketone body concentrations may not be enough if the cell's power plants, the mitochondria, can't efficiently utilize these fuel molecules. This insight opens up new avenues and questions about how we approach metabolic health and treatment strategies. The liver acts as a crucial energy hub in our bodies, storing and releasing fuel depending on what we need. In conditions like type 2 diabetes or metabolic dysfunction-associated steatotic liver disease, commonly called fatty liver disease, mitochondria within cells, these are the tiny structures responsible for energy generation, struggle to switch smoothly between different fuel sources. Researchers at the German Diabetes Center, DDZ, Heinrich Heine University Düsseldorf, HHU, and the University Hospital Düsseldorf, UKD, took a groundbreaking step by directly measuring how well mitochondria in key organs such as the heart, muscles, liver, and kidneys metabolize ketone bodies, small molecules produced by the liver during fasting, exercise, or low-carb diets. Ketone bodies serve as vital alternatives for energy production, especially when glucose levels are low. Normally, in healthy individuals, higher ketone levels can support the energy needs of the heart, skeletal muscles, kidneys, and other tissues. But their effectiveness hinges on the mitochondria's capacity to process these molecules. As Professor Michael Roden, the scientific director of the DDZ and leader of the study, explains, ketone bodies are more than just an alternative fuel, they play fundamental roles in energy production across organisms. Our research aimed to understand if mitochondria in individuals with diabetes or fatty liver disease can still harness ketone bodies efficiently. The key takeaway: simply raising ketone levels doesn't automatically translate into better energy generation in people with type 2 diabetes. In their study, researchers analyzed tissue samples from obese participants, some with and some without type 2 diabetes or fatty liver disease. Using a cutting-edge measurement technique known as high-resolution respirometry, they were able to directly assess mitochondrial energy output from ketone bodies, a feat unlike previous studies that only measured blood or tissue ketone levels. As Dr. Cesare Granata, senior researcher, notes, this method provides a more accurate picture of mitochondrial function related to ketone metabolism, although it captures data ex vivo, outside the living organism. The findings were quite revealing. Across all the insulin-resistant conditions examined, mitochondrial ability to generate energy from ketone bodies was diminished. Specifically, in overweight individuals with type 2 diabetes, the heart and muscle cells showed a significant decline in their capacity to utilize ketones for energy. Similarly, liver cells from those with fatty liver disease exhibited reduced ketone processing. Dr. Elric Zweck, the lead author, underscores the importance. This impairment was more pronounced than the general decline of mitochondrial function itself, indicating that ketone metabolism is especially vulnerable in insulin-resistant states. And this is the part most people miss. Improving mitochondrial function might be essential to restore effective ketone utilization, rather than just increasing ketone levels alone. What does this mean for future therapies? It suggests that strategies focusing solely on elevating ketone bodies, like fasting, ketogenic diets, or supplements, may fall short unless mitochondrial capacity to adapt is also addressed. Instead, therapies aimed at enhancing mitochondrial health and their ability to metabolize ketones could offer more comprehensive improvements in energy metabolism for those with diabetes and fatty liver disease. Further investigations are underway to decode the detailed mechanisms behind these metabolic alterations and identify targeted treatments to restore this critical aspect of cellular energy production. By expanding our understanding in this area, we might be able to develop more effective interventions that restore the metabolic flexibility essential for overall health. So, here's a question to ponder. Do you believe that boosting mitochondrial function could truly revolutionize diabetes management? Or are there risks in focusing too heavily on cellular energy pathways? Share your thoughts, and let's discuss where the future of metabolic therapy might be headed.